Hi guys, can you hear me? Hopefully the answer is yes. I know last week I forgot to go live and I was talking to myself and it was terrible, but hopefully you guys can hear me and welcome. Thanks for tuning in. So as a reminder, I'm Caroline from Fiveable. I'm an AP biology teacher teaching in Chicago, Illinois, and I will be instructing various topics for the next couple months leading up into the AP biology exam on Tuesday or Wednesday nights, usually Wednesday nights at 6 p.m. Central or 7 p.m. Eastern. Going through the scope and sequence of AP biology and the most challenging topics that could possibly be covered on your exam. So today you're in for a treat because we are talking about Hardy-Weinberg, which is oftentimes one of the harder topics that students have to kind of grapple with throughout the course of the school year. So hopefully you might have seen this during your first couple of weeks of school. If not, it will give you a really good preview as far as what you can expect to see, and you'll already be ahead of the game. Because again, it is one of those topics that tends to be more challenging, and it does show up on the AP exam every year without fail. So it's definitely a good one to know about. It especially shows up on those grid-in questions. If you're not familiar, there's always 6 math grid-in questions at the end of the AP exam, and this is a common target in those. Also been seen in free response questions, and multiple choice, so it's really fair game throughout the test. All right, so I'm going to hop over to a PowerPoint. Okay, I'll also make sure that I'll send this link out really quick. Here is the link to the PowerPoint in your chat box. If it doesn't let you click into it, I can show the change. Should be anyone with the link. Okay, perfect. Should be that anyone can open that, and I'll share it on my screen as well. And then we'll get started. I'll do a little review of genetics while we jump into our discussion today. So here we go. Okay. So as I said, we're talking about Hardy-Weinberg, oftentimes one of the most challenging subjects if you're just tuning in. Okay. First and foremost, I just wanted to go over a brief review of genetics. So this is really, really important when we're talking about Hardy-Weinberg. And if it's something that you haven't had as much of experience with in an intro biology course, it's really, really important that you have a crash course reminder of genetics. Otherwise, this Hardy-Weinberg stuff is going to be really, really confusing. So I have this picture on the slide that shows chromosomes with 2 different alleles, allele 1 and allele 2, and a couple of different vocab words that I want to go through. So first and foremost, an allele is just a version of a gene. So in this case, we can imagine that this spot on the chromosome that's highlighted light blue, where it's allele 1 and allele 2, is one single gene. So maybe it's the gene for eye color in flies, okay? And you can have different versions of that gene. There's a dominant version of that gene and a recessive version of that gene, in this case highlighted with the capital and lowercase a. So if you're looking at the chromosome that says allele 1, that's going to be, with the capital A, that's going to be our dominant version of that gene or that allele. And then on the right-hand side where you see the lowercase a, that's going to be the recessive version of that gene, okay? So because we are diploid organisms, diploid, di meaning 2, okay, and most of the organisms that we look at, most of the species that we study are diploid, so they have 2 chromosomes or 2 versions of each chromosome. We get all of these different genotypes, which you can see at the bottom of your screen. There's one that says homozygous, one that says heterozygous, and another one that says homozygous. So because you have a version of each chromosome from mom and a version of each chromosome from dad, making us those diploid organisms, as I just explained, you can get 2 dominant versions of that allele or 2 dominant versions of that gene, which you would see in the bottom left-hand corner where it says homozygous with the 2 double A's. That would be homozygous dominant, okay? Homo meaning same, okay? And then dominant meaning that you have 2 of those dominant alleles. The middle 2 options are showing you homozygous or heterozygous, individuals. So you can have a big A and a little a, or a little a and a big A, same thing, okay? But we call those heterozygous, hetero meaning different, okay? So you have 1 dominant and 1 recessive version of that gene. And then lastly, all the way to the right, there's homozygous recessive. So there's two little a's that this person inherited. Homo, again, meaning same, and they're recessive in this case because they have two lowercase a's. Okay. The last really important genetics things that you need to remember is the difference between a genotype and a phenotype. Okay. So a genotype, gene for genes, it's what your genes look like. So if you look at the bottom of this slide where it says homozygous dominant, heterozygous, and homozygous recessive, those are all genotypes. It's telling us what the genes of that organism are, okay? Phenotypes, I think phenotype, physical appearance, okay? So phenotypes are what the organism looks like physically, okay? There are only two options for phenotype. You can be dominant or you can be recessive. Okay, so again, hopefully this is review for most of you and you're like, oh my gosh, I already know this. That's great. I'm just giving you a little crash course reminder because if you don't know those things, Hardy-Weinberg gets really, really tricky just because you don't know what you're given and what you're looking for. Okay, so if you have homozygous dominant genotype or a heterozygous genotype, both of those species, as you can see, or both of those individuals, have at least one dominant allele. That one dominant allele will overpower the recessive allele, and therefore you will appear dominant. You will look dominant, so your phenotype is dominant, okay? So if we were talking about fly eyes, which I was mentioning earlier, and the dominant phenotype is having red eyes, then it wouldn't matter if your genes are homozygous dominant or heterozygous, you would still look like you had red eyes. So unless we tested your DNA, we wouldn't know if you were heterozygous or homozygous dominant, we would just know that you have red eyes. On the flip side, if you're homozygous recessive and you have two recessive alleles, this is the only time that we'll actually see a recessive phenotype or a recessive physical appearance, okay? So for flies, white eyes is recessive. So if you have two lowercase a's or two recessive alleles, then you would show that white-eyed phenotype, and we would look at that fly and say, oh yeah, that's homozygous recessive because it's showing the recessive phenotype, okay? So again, really, really important that you remember the difference between a genotype and a phenotype, and that you know that there's only two phenotypes, dominant and recessive, whereas there's three genotypes, homozygous dominant, heterozygous, both of which look dominant, and then homozygous recessive, okay? All right. That's your crash course in genetics. If I'm going too fast later and you need another little reminder, just type me a message and I'll make sure that I go back through it. But I'll just assume that everyone has a pretty good basis of this stuff from a previous course. Okay, the other really important word that you need to be familiar with that we talked about briefly last week if you tuned in is a gene pool. Okay, so a gene pool of a population, so remember a population is a group of the same species living in the same area, okay? The gene pool for that population is just if everyone threw their alleles for a certain trait into the middle and we just counted up how many dominant and how many recessive alleles we have belonging to everyone. So as you can see in my little cartoon example, if all of these pigs threw their alleles for a certain trait into a pond, it's not a literal pond, this doesn't actually happen, okay? But you would be able to count up all of the different alleles that make up all of that, the traits in that population. Okay, so we're going to be talking about the gene pools a lot when we're looking at Hardy-Weinberg and evaluating how a population is changing. All right, important vocab words. You have the PowerPoint link in the chat box, so you can look back through those if you're confused while I'm talking about the actual topic of today, which is Hardy-Weinberg. Okay. All right, so this is also a review from last week if you tuned in. But in order to understand why Hardy-Weinberg exists and why it's important that you're studying it, you need to know why alleles change, okay? So again, an allele is a version of a gene. We have two of them for each of our genes. They can be dominant, they can be recessive, okay? In a population like we were talking about, so same species and same area, the frequencies of the alleles or the traits in that population might change, okay? 5 reasons they can change. 1, natural selection. The environment changes and there's pressure on that population, okay? Then we may see some traits that are valued more than others. those traits are the ones that are part of organisms that are surviving and reproducing, and we have this shift in the alleles towards that one trait. Okay. Number 2, mutations. Mutations are something that we have absolutely no control over, right? It's a random change in the DNA. So mutation, if we randomly change DNA, we're randomly changing an allele. And if it's something that's beneficial, that organism might reproduce more, and we would see it more in our gene pool. Next one, gene flow. If you remember, I can grab my picture. Gene flow, we have one beetle coming into another population. So these are all red beetles. We're moving across into a population of mainly brown beetles. And because of that, we've increased genetic variation in this new population due to the fact that they now have a new trait, right? So that would definitely change allele frequencies. Next one, genetic drift. So 2 examples of that. That's the bottleneck effect and founder's effect. So again, this is review. Okay. With the bottleneck effect, that's when we have some mass catastrophe where only a few survive. Those are the ones that are replicating, and we have a less diverse population as a result of it, because a lot of our traits have died and washed out. Okay, so that's an example of a way to decrease some alleles in a population or increase others. And then founder effect is when we have one subset of a larger population that moves somewhere else and starts a new colony. So something like moving to an island or moving across a geographical barrier. And then we have that small subset that would have a different allele frequency than the starting population. Okay. And then lastly, we talked about this last week, but non-random mating, the biggest example of this is sexual selection. So actually selecting a mate based on different traits can change different allele frequencies, right? So if birds decide that they like, if female birds decide that they like longer-tailed male birds, they're going to mate with the longer-tailed male. Oh, sorry, Tanea, thanks for the suggestion. But if birds decide that they're going to mate with the longer-tailed male birds, then we would see a shift towards that longer tail, okay, which would change the allele frequencies. We would have more of those longer alleles in our population, okay? So all of those things can change allele frequencies. Why in the world am I reviewing all of this? Well, because it's necessary in order to understand what Hardy and Weinberg came together to create when they came up with Hardy-Weinberg Equilibrium, okay? So the reason that this equilibrium, so first of all, equilibrium means like everything is equal. We've maintained homeostasis. Nothing is changing, okay? And basically, Hardy and Weinberg came together and they said, okay, how can we determine if what a population would be completely at rest if it was not evolving, okay? You might be wondering to yourself, why in the world would I want to be looking at a population that's not evolving in my evolution unit? Well, the basis of this is that if you can look at a population that's not evolving, then you can compare a current population to that to see how much it's changing or to see how much it is evolving, okay? For reference, if I wanted to know how much growth you gained on the SAT, let's say, I would need to know where you started at in order to determine how far you grew. You could tell me you got a 1280 and I'd be like, wow, that's really great. But if you started at a 1270, right, then that doesn't mean much to me, okay? So Hardy-Weinberg Equilibrium will tell us the baseline if this population were not to be evolving, okay? And then when we compare it to an actual population, we can see how much that population has evolved or how quickly it is evolving, okay? So that's Hardy and Weinberg's goal. So the next slide is really, really important. It's the 5 things that must be true in order for Hardy-Weinberg to be in equilibrium. So again, if these are the 5 things that are necessary for alleles to change in a population, then Hardy and Weinberg just said, okay, if a population is going to be in equilibrium then or not be evolving, then all 5 of those things need to not be happening. That's a lot of double negatives, but let me show you and it will make more sense, okay? So Hardy and Weinberg said, okay, for a population to not be evolving, these 5 things would be true. 1, there would be an incredibly large population. Why do we need an incredibly large population? So that we don't have genetic drift, okay? Remember with bottleneck effect and founder effect, that happens when we have a really small population size that's not comparative to the original population. So for Hardy-Weinberg to exist, or this equilibrium to exist, we need a very large population. 2, mating is completely random. Whoever you bump into is your mate. Okay, there's no selection whatsoever. It's just completely random, and whoever you see, whoever you bump into, that's the person that you're going to mate with, or the organism that you're going to mate with. I know that sounds crazy. 3, there will be absolutely no mutation in these populations. Okay, no DNA will be mutated. 4, no immigration or emigration. Nobody's coming in, nobody's leaving. There's no gene flow in this population. And 5, there's no natural selection. So the environment is not changing. Okay, there's no environmental pressure on this population that's going to put stress on it to survive and reproduce. Okay, so those are the 5 things that are completely the opposite of what would change alleles, right? And so Hardy and Weinberg came together and said, if these 5 things happen, there will be no evolution. Do these 5 things ever actually really happen? No, not really, okay? Very, very infrequently, maybe in some isolated population somewhere, okay? But for all intents and purposes, we're just using this as a baseline to see how quickly a population is evolving. So that's the theory behind it. Now we're actually going to look at the equations, which is probably what most of you are waiting for, okay? And I'll be able to show you some practice problems, which is the most helpful thing here, right? I can talk about Hardy-Weinberg as much as I possibly want, but it's really hard to learn unless you're actually doing the math. So if you don't already have a calculator with you and like a pen or a pencil and some paper that you can write some things down, you definitely want to get those things because Hardy-Weinberg definitely requires the calculator, and I want you to be able to be practicing alongside of us to make sure that you're getting the right answers. So go grab that now if you don't have it already. All right. Here they are, the Hardy-Weinberg equations and definitions. Okay, so there are 2 equations that can be used for Hardy-Weinberg equilibrium. One, the first one is nice and simple. It's P plus Q equals 1, and I'm going to define all those variables for you in a second. The second one you'll notice is just a perfect square of the first one. Okay, that's where we get P squared plus 2PQ plus Q squared equals 1. Okay, what does P mean? What does Q mean? P squared, blah, blah, blah. What does it all mean? Well, that's defined for you below. And on the AP exam, you would get both equations as well as the definition of P and Q on your equation sheet. Okay, so these are not things that you need to memorize. You just need to know how to use them. Okay, all right. So P and Q, focusing on that first equation. So that first equation is used to look at allele frequencies and how they're changing. Okay, how they're changing in the gene pool. So you're gonna have to bear with me here. I'm gonna explain this a couple times because students always mess it up, okay? But P and Q are going to be equal to the frequency of the dominant and recessive alleles in a population. Okay, so P, I think of P as in powerful. P is the frequency of the dominant allele, okay? On the flip side, I think of Q as in quiet, and Q is the frequency of the recessive allele, okay? So remember, an allele is just 1/2 of a whole. It is one part of an entire individual. You have 2 alleles for each trait that you have. Okay, so I'm going to grab my poker chips. So if you're homozygous dominant, you have 2 dominant alleles. If you're heterozygous, you have a dominant and a recessive allele. And if you're homozygous recessive, you would have 2 recessive alleles, okay? But just a single allele is what we're going to be counting in the allele frequencies for P's and Q's, okay? I'm going to show you an example picture in a second. But when you're thinking about P's and Q's, you just want to be thinking about individual alleles or individual versions of genes and not whole organisms, okay? That's going to become really, really important when we take a look at some of these practice problems and you're trying to decide what you're given and what you're looking for. Okay, more on that in a second when I give you a sample picture. Okay, other things that are defined below are helpful and they're not given to you on the AP exam, so it is something that you need to remember. Okay, so these next 3 things define the second equation and what that equals. Okay, so we have P squared, that's 2 powerful P dominant alleles. Okay, and that's going to be equal to the frequency of the homozygous dominant genotype. Okay, So P squared is our frequency of all of the homozygous dominant individuals in our population. 2PQ is the frequency of the heterozygous genotype. They have one P and one Q, right? That's why we call them heterozygotes. So those would be all the individuals in the population that have one dominant and one recessive allele, okay? Both homozygous dominant and heterozygous P squared and 2PQ are going to look dominant. They're going to have the dominant phenotype. Okay, so for my example in the beginning of this podcast, okay, that would be the red-eyed flies. So if I tell you that you have a certain number of red-eyed flies, that's going to be a combination of homozygous dominant and heterozygous individuals. Okay. Lastly, Q squared, 2 recessive alleles, is going to be equal to the frequency of the homozygous recessive genotype, or how many organisms we have that are homozygous recessive. Okay. The really great thing about Q squared, and the reason it's going to become your favorite of all the variables, is because if something is homozygous recessive, it's the only time that that thing will look recessive. So from my example in the beginning, if I told you the number of flies that have white eyes is the recessive phenotype, okay? So that frequency would be the same as the frequency of Q squared, okay? Which is something that makes the recessive phenotype different from the dominant phenotype, okay? This is gonna make a lot more sense once we start practicing, I promise, and when I show you this next picture, okay? But something I want you to notice about these equations is that they always equal 1, okay? So that means our frequencies are decimals. They're parts of a whole, okay? So if I was trying to take a look at a population and I was trying to count up the number of dominant and recessive alleles in that population, let's say we have one homozygous dominant individual and one heterozygous individual in our population, okay? If I just want to look at allele frequencies, I would have 3 dominant alleles out of 4 and 1 recessive allele out of 4, okay? So P would be 3 out of 4, or 0.75, and Q would be 1 out of 4, or 0.25, okay? And if you look at equation 1, 0.75 plus 0.25 is equal to 1, right? Basically, it's a fancy way of saying if 75% of my alleles are dominant, then the other 25% are recessive, right? It's just making sure that all of our alleles or all of our genotypes are accounted for, okay? Equation 1 is really the only equation that you'll use. Equation 2 is more just there just to define the frequency of the homozygous dominant, heterozygous, and homozygous recessive genotypes, okay? All right, I'm going to show you a picture where we can practice kind of calculating the difference between P, Q, P squared, 2PQ, all that nonsense, okay? And then we're actually going to jump into some practice problems where you guys will be able to see what this looks like in practice. Okay. So we're looking at these little creatures. Let's just call them sheep because that's my last name, and so I'm very used to dealing with sheep, okay? And you'll notice that we have some males and some females that look different and have different genotypes below them, okay? So the first thing that you should notice is that all of the black males and females are what we would call heterozygous. They have 1 dominant allele and 1 recessive allele, okay? If we were just looking at the sheep, we would just see, hey, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 black sheep, okay? But because we also have their genotypes, we can say that there are 12 heterozygotes in this population, okay? On the flip side, we have 12 homozygous recessive white sheep, okay? Even if I didn't see the 2 little b's below them, if I knew that white was the recessive phenotype, I would know that all of those sheep are homozygous recessive because that's the only way that they would look white, okay? So that's something important to note from the beginning. So if I were to look at this population, in total I have 1, 2, 3, 4, what did I just say? 24 sheep. So I'm gonna flip to a screen really quick. You can, remember you have this PowerPoint that you can pull up on another screen if you wanna follow along with that. I'm gonna pull up a whiteboard screen very quickly here to show you, hopefully. All right. Should be able to see a whiteboard now. Okay, perfect. So to show you, so we have 24 sheep. in that example. Sorry, you have to bear with me. I don't have a mouse, so I'm trying my best right now. Okay, we have 24 sheep in that population. We have 12 black sheep, and we have 12 white sheep. Okay? So the phenotypes, we're 50-50, right? We have 50% of our sheep look black, 50% of our sheep look white, so 50% dominant, 50% recessive phenotype. Okay? Now with this, I can take this a step further because it told me the genotypes as well. Okay, so I have 12 sheep that have 2 little b's. Oops, I almost tried to make an A because that's what I usually write. 2 little b's, okay? And my black sheep, I have 12 that have 1 big B and 1 little b, okay? So if I was trying to set these equal to variables from Hardy-Weinberg, okay, what I would do, ooh, that's not what I'm, there we go. Learning my black word. Thanks for bearing with me. Okay, what I would do is I would tally up how many alleles we see. So you could just count. If you're on your screen and you're looking at the sheep, you can just count how many big B's and how many little b's you see, and we can sort it that way, okay? But if I want to see how many dominant big B alleles I have, that would be equal to P, the frequency of the dominant allele, and how many lowercase little b's I have. We can count them, or we can do some simple math here. Okay, so our 12 black sheep, they're going to have 12 big B's, right? Because they each have 1 big B, and they're going to have 12 little b's. Okay, our 12 white sheep are going to have 2 times 12, or 24 little b's. Because remember, they have 2 versions of each, but theirs just all happen to be little b's. Okay, so my 12 big B's, I can write up here. And my 24 plus 12 little b's will give us 36 little b's, make up the entirety of our population. So if I wanted to change this into a frequency, right, which is what P and little b would be Q are equal to, okay, I need to make this the 12 big B's that we have, over the total amount of alleles that we have for our population. So if we have 24 sheep and they each have 2 alleles, then we have 48 alleles total. So I would put 12 over 48, and I would get that 1/4 or 0.25 of my alleles are dominant, and that's equal to P. Okay? If I put 36 over 48, then I would get 0.75, or 75% of our alleles are recessive, which would be equal to Q. Okay, so the frequency of the dominant allele P is equal to 0.25. The frequency of the recessive allele Q is equal to 0.75. Okay, I can also look for the frequencies of the heterozygotes and the homozygous recessives in the population. So 2PQ is what we use to represent heterozygotes. You just have to remember that, okay? If you look back on the slide before, you'll see that, okay? And that's going to be equal to, we had 12 of them, right? 12 heterozygotes, all of our black sheep out of 24 total sheep. So 0.5 would be equal to 2PQ as a frequency. And then Q squared would be equal to 12 out of 24 as well. So we had 12 homozygous recessive sheep, or 0.5. Okay, so that's how we could define all of those variables with a given population, knowing what they look like and what their genes say. Now, usually we don't know what they look like or what their genes say, okay? And we're going to see this in the example we take a look at in a second, okay? And then we're going to have to do something a little trickier. That's where Hardy-Weinberg gets a little tricky. But if you can just break down the difference between an allele, a genotype, and a phenotype, you're already in really good shape for Hardy-Weinberg, okay? So we're going to jump into a practice problem. I'm going to guide you through a few, and then hopefully if you have paper with you and a calculator, we can do a little bit more of a guided practice where I'll let you work for a second and then we can circle back and I can run through how to do the problem and you can see how you did, okay? Practice jumping in is really the best way to take a look at these problems. Otherwise, you're going to have a hard time understanding them. So I'm going to erase what I just wrote. And then we're going to hop over to our first practice problem to see what these actually look like. Okay? All right, I'm going to switch the screen on you for a second. Boop. Okay. Here's our problem. So in a certain flock of sheep, stayed with sheep again, okay? In a certain flock of sheep, 4% of the population has black wool and 96% of the sheep has, that should say, white wool. Okay, so 4% of the population has black wool, 96% of the population has white wool, and we are going to assume that the population is in Hardy-Weinberg equilibrium. Okay, if black wool is a recessive trait, okay, ding, 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 so the black wool population is recessive here. What percent of the population is heterozygous for this trait? Okay, that's a lot of words. Your first step always for a Hardy-Weinberg problem is to figure out what is given, okay, and what you're looking for. So I call that the given and the unknown, okay? What the heck are the numbers that they gave you, and what the heck are you looking for in the problem? If you can figure those two things out, and you know how to square and square root, then you know how to do Hardy-Weinberg, okay? So let's figure out our given and our unknown in this problem. All right. So given, so this is where we're going to look at the numbers. So if you reflect back on that problem, looked like someone didn't have access. Let me really quick check the settings on this document. All right. Hopefully that helps. Did that help anyone? Okay, just bear with me. You should be able to see the problem, so it shouldn't be that big of a deal. Okay, back into it. So, reminder, 4% of the population had black wool, 96% of the population had white wool, okay? So, what we need to know is then what that actually means for us. So, 4% of the population has black wool, which is recessive, and 96% of the population has white wool, which is dominant. We're going to see this in another problem, but for the sake of an easier start, just know that we don't want to deal with that dominant population because we don't know if they're homozygous dominant or heterozygous. Remember that these organisms, homozygous dominant, and these organisms, heterozygous, they all just look the same. We don't know what their genes say unless we're going out to all these sheep and drawing their blood, which we are not, okay? So that 96% of sheep that had white wool could be either of these things. It's some sort of combo of homozygous dominant and heterozygous individuals. So we don't know much about them. So we are going to ignore them. The thing that we do know, though, is that the 4% of the population that is black, they gotta be homozygous recessive. Otherwise, they're gonna look white. The only reason that you have that black wool is if you are homozygous recessive, okay? Remember, homozygous recessive, if you flip back a few slides, is equal to Q squared, okay? That is just always true, no matter what. And I'll try to, if people don't have access to the document, give me 1 second. I will copy that into our chat. That way you have it. Perfect. Okay. All right. Continue chat now. so that you can see the equations and so that you can see what p squared, 2pq, q squared, p, and q all mean. Whew. Okay, so knowing that 4% of our population looks recessive, that means 4% of our population is homozygous recessive. And so we can set that 4% equal to q squared, right? So 4%, changing that into a frequency, I make that 0.04% of the population is homozygous recessive. That's equal to q squared. That's what's given to us in the problem. That's what's usable in the problem, right? The 96%, ignore it. What are we looking for here? So if you read this question over again, if black wool is a recessive trait, what percentage of the population is heterozygous for this trait? If you look at the things that I just listed in the chat for you guys, okay, you'll notice that heterozygous, what we're looking for is 2pq, okay? So the value of 2pq is our goal, what we're going to find, and we have q squared being equal to 0.04. Okay? Finding the given unknown and unknown first is essential to understanding these problems, because now we know what numbers we have that are usable, and we know what numbers we need to find or what variables we need to find, and then the rest is pretty simple. Okay? So next, I'm using a method called the guess method. So we found the G, we found the U. The next step is to look back at your equations, which I just posted in your chat. Okay, but equations, or E in the guess method, the equation that you're always going to use is P plus Q equals 1, okay? Because if you can use P plus Q equals 1, you can find P, you can find Q, and therefore you can find any of the genotypes that you might need to find, okay? All right, so we're given Q squared, but I need to use this equation P plus Q. Any ideas of how we could go from Q squared to something that might be usable in the equation P plus Q equals 1? No? No one's with me anymore? We got to square root it. So this needs to be something that you become really familiar with. Yes, thank you, Tania. So we need to square root this. That's how you get rid of a square, right? It's just like the inverse function of a square, okay? So if we have q squared and we need to find q so that we can plug it into our equation, then you need to grab your calculators and square root 0.04, okay? And you can write this out. It's hard for me to write on the whiteboard and switch back and forth into PowerPoint. I'll try to do that for some of our next problems coming up, okay? But for all intents and purposes, you can just grab your calculator, you can square root 0.04, okay? And you should get that Q is equal to 0.2, okay? So Q squared is equal to 0.04, therefore Q is equal to 0.2. We have this equation, P plus Q equals 1. Okay, so now we can find P, right? If P plus Q equals 1, P plus 0.2 is equal to 1 then. And if you think about it logically, because some people just like to actually think about what they're doing, okay? If 20% or 0.2, right, 20% of your alleles are recessive, then the other 80% must be dominant. Otherwise, we have aliens in our presence or something crazy is happening, and that's just not happening. So 20% is Q, then 80% better be P. And if you do the simple math there in P plus Q equals 1, you will find that to be true, okay? So P is equal to 0.8. Q is equal to 0.2. If your P's and your Q's don't add up to 1, you did something wrong. So back up, see where you messed up, and try again, okay? All right, so last step here is to solve. So remember, you may have forgotten when we're on this like winding road to nowhere right now, what we're actually looking for. And what we're actually looking for, unknown if you go up to U, is the percentage of the population that's heterozygous, okay? Remember from looking at the chat, heterozygotes, the frequency of them is equal to 2pq. And now we have P and we have Q. So this just becomes the simplest math problem ever, okay? So we can plug in for P and Q, okay? And we will get 2 times 0.8 times 0.2, which is equal to 0.32, okay, or 32% of the population being heterozygous, okay? So to back up or rewind what we did there, we found what we were given. We were given that 4% of the population was homozygous recessive or looked recessive, which you know as brilliant biology students means that they're homozygous recessive. We used that to find Q squared, okay? We knew that we were looking for 2PQ because the question is asking us the percentage of the population that's heterozygous. We know that we're going to use equation 1, P plus Q equals 1. So to make that usable for ourselves, we found Q from Q squared by square rooting. We plugged into P plus Q equals 1 to find P, and then we plugged into 2PQ to find the percentage of the population that's heterozygous, okay? So the easiest way to do these, again, is to figure out what you're given and what you're looking for, and know that you need to use P plus Q equals 1 and figure out something that's going to make that usable for you, okay? All right, let's do some more practice problems so that we can see, because the biggest challenge with Hardy-Weinberg is that what you're given and what you're looking for changes every time you do the problem. Okay, so whenever I talk to math teachers about how to teach Hardy-Weinberg, blah, 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 they're like, ooh, you just need to keep practicing with that because there's a bunch of different ways that you can set this up. And the best way that you can do it is just to continue to practice. Okay, so let's do that. So my first question, all my questions are either like puppy or Harry Potter related, just because that's what I'm into. So you just got to bear with me. Okay, but our first practice problem that we'll look at together, I guess it's really our second practice problem, is going to have to deal with puppies. Okay, so I have, you'll see given unknown equation, substitute and solve written up here. Okay. So if you have a separate piece of paper and a calculator, you should write this down, given unknown equation, substitute solve, or just write GUESS so that you can follow along with us in this method, okay? All right. In a population of puppies in Hardy-Weinberg Equilibrium, the frequency of the dominant allele, bells should be going off in your head, is 0.6. If brown fur is dominant over spotted fur, how many of the puppies would you expect to have spots? Whew, okay, lots of information. So again, we need to break this up into what we're given, what we're looking for, and then what we can use this for, okay? All right, so I have that the frequency of the dominant allele is equal to 0.6. 0.6. Anyone know what that would be equal to? What variable? The frequency of the dominant allele? You can look back in the chat for help. Should be P. So if you look into the chat, P is equal to the frequency of the dominant allele, and that is the specific words that are used in the problem. It doesn't tell me that 0.6 or 60% of the population is brown or looks dominant, or that it's homozygous dominant. It's just telling me that the amount of dominant alleles in this population is 0.6 or 60%. Okay, so that's my given. I'll write that on my whiteboard screen in a second. Okay, but I don't want to get rid of the problem yet. My unknown then is how many of the puppies would you expect to have spots, okay? So that's the amount of puppies that look recessive. A lot of my students are like, oh, okay, recessive, that equals Q. No, it doesn't equal Q. It doesn't say how many alleles in this population are recessive. It says how many whole puppies would you expect to have spots, okay? So if a puppy looks spotted, it shows the recessive phenotype. That means it's homozygous recessive. So we're looking for Q squared, okay? So I'm going to switch over real quick. You're going to lose the problem. Hopefully you have that written down on a piece of paper. Switch over to sharing my whiteboard with you. Good. Okay, perfect. And I'll start writing out what we have been given and what's our unknown, okay? So our given, we just went through this, is that P, the frequency of our dominant allele, is equal to 0.6. That's what's in the problem, okay? Our unknown, what we're looking for, is the number of spotted puppies. Those are all the homozygous recessive puppies. So we're looking for Q squared. Always double check these. Don't make silly mistakes like setting that equal to Q or something, okay? And the equation that we're going to use, as always, is P plus Q equals 1, okay? That will always be given to you on the AP exam, but it's also really easy to remember. And if you practice these enough, it will be ingrained in your brain for all of eternity. Okay. Oh, I don't want to drop a file. Ah. Stop. Okay. That looks funny on your screen too. I don't know why that happened. All right, here we go. So we're going to substitute in then. So we have P. We know we're going to use this equation P plus Q equals 1. So this is actually an easier start than the problem that we just did because we don't need to square or square root anything. We're given P. We can plug it right in. So I'm going to start substituting. I'm going to substitute in for P, 0.6 plus Q is equal to 1. Okay, and again, if 60% of our alleles are dominant, then immediately in your brain you should be thinking, oh, then 40% of our alleles are recessive. Okay, but by doing some simple math and subtracting 0.6 from both sides, you'll get that Q is equal to 0.4. Easy enough, right? 40% of our alleles are recessive if 60% of our alleles are dominant, right? Last step then, go back and look at your U. What are we actually looking for here? Oh, we're looking for Q squared because we're looking for how many puppies have spots, right? We have Q, we need Q squared. Easy solution, right? We just need to square it, okay? So it's the same thing as like plugging Q in up here to our unknown. So we're looking for Q squared. We have Q. We can square it. And if you square 0.4, you should get 0.16, okay? Which would mean that 16% of our puppies would have spots, okay? And that would be the answer to your question. If you're wondering like, hey, how much do I need to round this, etc., you're just not quite sure. On the AP exam, they will always tell you, okay, how far you should round it. But a good rule of thumb is to always round to the nearest hundredth, just because you're dealing with percentages here, okay? So 0.16 is 16%. You wouldn't want to round that to 0.2 because that would be 20%. That's pretty substantially different, okay? So keep it with the hundredths if you're dealing with that. Sometimes they'll actually ask you to give like to the nearest tenth of a percent, in which case this would be like 16.0%. Okay, so sometimes they'll do that, but they'll always tell you in the problem what you should round it to, so no worries there. All right, looks like we might have time for one more guided practice problem. Wanted you guys to do some on your own, but they just take a long time, and the rationale behind them takes a while, so we'll have to do some more practice during our review at the end of the year. Okay, so I'm going to erase this. We're going to jump into one more practice problem. I'm actually going to skip problem 2, even though it's great. Look at that picture of a hippogriff, so majestic. Oh, you can't see it. Oh, so you don't even know how majestic it is. Give me a second. I'll fix that for you. None of my students like Harry Potter, and it's very upsetting to me. So hopefully some of you like Harry Potter because it's truly like my #1 passion in life. Maybe that's just me. All right, so we're going to skip over problem 2. Now you can see the hippogriff. You see what we're missing. But problem 3 is a little bit trickier, and I'm going to explain why. Okay? And I want to make sure that you see a problem that's set up like this before I leave you today. Okay? So problem 3 says, in a population of wildflowers, 72% of the flowers are purple. Purple is dominant to pink. What percentage of the population is homozygous dominant? Okay? So when I give this to my students each year, they're always like, oh, Miss Sheep, you gave us the answer in the question. 72% of the flowers are purple. Purple is dominant. 72% are homozygous dominant. I'm always like, no, that's not true. Because remember that 70% of flowers that are purple is just a mishmash of homozygous dominant and heterozygous individuals. I don't know which one of those purple flowers is homozygous dominant or heterozygous. It's just a mix. I wouldn't know unless I did a DNA test on them, okay? So we know nothing when we are given the percentage of the population that is dominant. We just don't know anything because we don't know anything about their genes, okay? This is different than when we're given the percentage of the population that's recessive because we know they're homozygous recessive. We know they're Q squared. We know exactly what their genes say, okay? So there's an easy fix when you're given a problem like this, okay? When you see that 72% of the flowers are purple immediately in your head, you should be like, that's not usable. I can't use that. That's a mishmash of p squared and 2pq. I don't want to touch it, okay? What you're going to do is solve for the amount that are recessive. So if 72% of your flowers are purple, then the remaining flowers must be pink, right? So if you have a total of 100%, 100 minus 72, 28% of your flowers are pink, okay? Pink is something that is usable because we know, again, their genotype is homozygous recessive or Q squared, okay? When we're talking about purple flowers, it's a mix of these guys. We don't know anything about their genes, so we can't solve for anything, okay? But we can solve for the fact that 28% of the population is pink. And I'm going to write that in a second. And we're looking for homozygous dominant or P squared. All right, I'm going to switch back to my whiteboard, and then this will be our closer for today. Okay, so my given. Ugh, I keep messing this up. I'll get it eventually. Our given is that 0.72 or 72% of our population is dominant. So I'm going to write this up here for you just so you don't forget what it means. That is a mix of p squared and 2pq. So I'm going to write it as p squared plus 2pq. We don't know how many of those are p squareds and how many of those are 2pqs. We just know they look purple, okay? This leads us to believe, right, if we do 100 minus 72, and we have 28% that are pink, and that's usable. Ooh, gosh, writing on this. People probably can't even read what I'm writing, but builds character, right? So 0.28 or 28% of our population is going to be equal to Q squared. Because they're recessive, they must be homozygous recessive then. That's the only way you look recessive, okay? And we know that homozygous recessive is equal to Q squared. Whew, good start. Okay, we're looking for P squared. We're looking for homozygous dominant. That was listed in the problem if you forgot, okay? But that's what we're looking for. We have Q squared, we're looking for P squared, and we somehow need to use our lovely favorite equation, P plus Q equals 1. Simple algebra. So that's the plus. Okay. All right. Here we go. I'm gonna zoom this and move it over a second. I'm getting real fancy here. All right. Now we'll jump back into drawing. So I know that I need to use this equation P plus Q equals 1, and we've talked about this now, right? You're always going to use it. I have Q squared though. Okay. So if I'm going to make Q squared something usable that I can plug into this equation, I'm going to need to go back and square root that so that I can find Q, okay? The way you get rid of a square is with a square root. So if I have Q squared is equal to 0.28, I need to go back and I need to square root both sides of this equation so that I can get rid of that square because I can't use it and figure out what plain old Q is. So if I square root 0.28, grab my handy dandy calculator. You should get 0.53, 0.53. So again, I'm rounding to the nearest hundredth because we're dealing with percents. 0.53 or 53% of our alleles then are recessive, okay? Now I have something that I can plug into this equation so that we can eventually find P squared, okay? So I'm going to substitute Q into our very simple P plus Q equals 1 equation. And I will get P plus 0.53 equals 1. All right. So again, if 53% of our alleles are recessive, the rest better be dominant. That's all this equation is telling us. Okay, so some simple math, subtracting 0.53 from both sides would tell me that P is equal to 0.47. If 53% of our alleles are recessive, 47% must be dominant. There's no in between. They're either dominant or recessive. Okay? Now I have P. This is where you say, okay, what do I do next? I don't know. What are we looking for? Go back to your unknown. I'm looking for P squared. I have P. Easy fix, right? So now we can substitute in to our original goal of P squared. And then we can just square 0.47. Then we have our answer. Okay? So it is truly life-saving just to find what you're given and what you're unknown, and know that you're always going to use P plus Q equals 1, because then it just becomes a couple of different challenging problems. And once you figure out the like 4 different ways they can ask these, you'll be a champion. Okay? All right. So you should get, when you square 0.47, you should get 0.22, or 22% of the population is homozygous dominant. So if you had incorrectly assumed that those 20% of the flowers that were purple were all homozygous dominant, you would be off by 50%, because actually only 22% of them are homozygous dominant. The rest are heterozygous. So don't forget about our heterozygous carrier friends. It's super important. And yeah, if you forget about them, you're going to get a whole lot of problems wrong, and nobody needs that. Okay? So let's jump back. That's going to be all the time we have for today. So hopefully you learned a little something about Hardy-Weinberg. Maybe it reinforced something. Maybe it introed something that's new and different for you. But the bottom line is you need to know your P's and your Q's, and your P squareds, 2PQs, and Q squareds. Okay? And if you break it down into given, unknown, equation, substitute, and solve, you'll be in much better shape, because you'll know what you have, you'll know what you're looking for, and you'll know how to make it usable using the equation P plus Q equals 1. Okay, the more practice you do, the better. So look for practice problems, look for the answers, tune into review sessions with me, and definitely follow us on Crowdcast, which is the platform that you're using right now. Follow Fiveable on Crowdcast and on YouTube to look for a lot more updates. As a reminder, the whole month of September is going to be free for you guys to tune into my live session. So next week, we're going to be talking about macroevolution. I think the last week, this month, we're talking about the origin of life. Okay, so all sorts of evolution topics this month. Stay tuned. I look forward to seeing you next week. Tell your friends to join us, and I'll see you next time. It'll be next Wednesday at 7 p.m. Eastern, 6 p.m. Central. See you guys.